All right, we are on Unit 6, Topics 7 and 8. So, mutations in biotechnology. This is one of my favorites. I know I say that every time, but for real. So, let's first talk about mutations. Mutations are changes in a genetic material of a cell. It alters phenotypes. Sorry, I'm going to go shut the door. I'm at school right now. Great. So, we know that genetic material, genes, code for phenotypes. Phenotypes are the outward expression of something. It's like the way an organism looks. Primary source of genetic variation, right? So mutations from one organism to the next, that's sort of where we get our genetic variation from. Normal function and production of a cell of cellular products is essential. We know that. So if something is to be mutated, it may lose its function. And then any disruption can cause new phenotypes. Changes can be large or small. Large scale changes are things like chromosomal changes, and small scale changes are like nucleotide substitutions, insertions, or deletions. Large scale changes have to do with non-disjunction and that sort of thing that happens during meiosis and mitosis. The small scale mutations here can happen during DNA replication. So small scale mutations, let's talk about them. One is a point mutation, so it's a single gene, it's a single nucleotide pair is changed or mutated. Substitution is the replacement of one nucleotide and its partner with another nucleotide pair. So for example, we had A, T, now we're going to change it, substitute it for G, C. So we just substituted it. Next, or a type of substitution mutation is a silent mutation. It still codes for the same amino acid. Remember the redundancy in the genetic code, that codons, there may be multiple codons that code for the same amino acid. So if we coded for something, or if we mutated something that codes for the same amino acid, nothing's going to change. It's going to be a silent mutation. Next, we have a missense mutation. So this is a type of substitution mutation, and it occurs when it results in a different amino acid. So substitution mutations, it's a type of point mutation, and it can either be silent or missense. Silent, no big deal. It's all the same stuff. We don't even see it. Missense mutations are a different amino acid entirely, and that could lead to the amino acid not folding properly. And of course, if we don't have the right structure, then it's not going to have the right function. Pause here, take notes. Oh, just kidding. Finally, we have nonsense. A nonsense mutation results in a stop codon. So if there is a mutation that occurs that codes for a stop codon where an amino acid really should have been going, then it's going to result in a shorter chain of amino acids, and again, possibly a non-functional protein. Now you can pause to take notes. So here's kind of the, a good overview of it. Let me there we go. Just make sure. So here it is at the DNA level. So we have T, T, C. So there's no mutation there. We see that the mRNA is the opposite. T goes to A, C goes to G. Great. And on a protein level, that codes for lysine. No mutation. Lovely. So now what we did was we substituted this guy, right? So it's no longer a C, now it's a T. The mRNA level looks like this, but because of the redundancy in the genetic code, it codes for the same amino acid. No problem. So even though there's a mutation, the final result is the same, so it really doesn't matter. All right, a nonsense mutation is something like this. So we are now going to mutate this thymine to this adenine. When we do that, the mRNA molecule here codes for a stop codon, and where lysine was supposed to be, now it just stops. So shortened amino acid chain, non-functional protein. Great, so now we have a missense mutation up over here. Don't worry about conservative or non-conservative, just know that there's a mutation that occurs here. You can see the mRNA chain right over there on the mRNA. This one codes for arginine, something entirely different. And THR, I forget exactly what that is, codes for something entirely different. So missense, no good. Nonsense, no good. Silent, meh, no big deal. Great. Pause here to take notes. All right. Another type of mutation that can occur, as opposed to mutating a base pair, is what we call a frame shift mutation. So when the reading frame of the genetic information is altered, this is disastrous. It has awful effects on the protein. So an insertion is when a nucleotide is inserted, and a deletion is when it's removed. Remember, we have here triplets in the genetic code. And if we inserted one, it makes the sentence all weird. If we took one away, same thing. So frame shift mutations changes the reading frame. So you can see here, sertraline, valine. I forget what that is. Now, the first two are fine, but the last two are completely wrong amino acids. So that's, again, a non-functional protein. Disastrous. Let's talk large scale. Oh, I'm sorry. Pause here to take notes. Let's talk some large scale mutations. So these are mutations that affect chromosomes. Non-disjunction. These are when the chromosomes don't separate properly in meiosis. It results in an incorrect number of chromosomes. The perfect example of this is Down syndrome. That's when you have 3 chromosomes at location 21, and it's caused by non-disjunction in meiosis, right? So you have the chromosomes lining up here, the metaphyseal plate. They go in opposite directions, except this one accidentally goes over there, and we end up with 3 chromosomes in that one. So it really is, it's just a pulling in the wrong directions. Non-disjunction, we call it. Next, we have translocation. That is when one segment of a chromosome moves entirely to another. It is possible. It happens. An inversion is when it's reversed. A duplication is when it's repeated. And a deletion is when it's lost. Okay, so we're taking a whole segment of one chromosome, and it can happen similar to non-disjunction, where this portion gets pulled over here. You know, maybe it doesn't align right at the kinetochore. Number of things can go wrong. Inversion is when it's reversed, duplication is when it's repeated, deletion is when it's lost. Pause here to take notes. Great. So anytime mutations occur, we're subject to natural selection. Genetic changes can sometimes enhance the survival and reproduction of an organism. This is kind of slowly easing us into our next unit, which is natural selection and evolution. But this is why survival of the fittest is so ridiculous, is that they're not trying to, just the phrase, not the theory. The theory is correct, but it's not survival of the fittest. It's mutations happen, and when they're good, yay. And when they're bad, boo, right? Like, it's not like it's throwing spaghetti at a wall to see what sticks. Maybe the mutation will be passed on to the next generation, and maybe it won't. So, natural selection. Great, let's talk about increasing genetic variation. So prokaryotes exchange genetic material through horizontal genes transfer. You guys did that in the lab with the E. coli. You horizontally exchange genes from technically one bacteria, because that plasmid didn't come out of nowhere, into the E. coli itself. So if there's a mutation that's beneficial, then it can also be transferred. Transformation. So we did bacterial transformation of E. coli in the lab. It's just uptaking of DNA from a nearby cell. We have transduction is also another form of horizontal gene transfer. It is how viruses transmit genetic material. Conjugation is a cell-to-cell transfer of DNA. We saw that with things like quorum sensing, as you can conjugate a piece of DNA from one cell to the next. And transposition, which is the movement of DNA segments within and between DNA molecules. So these are all in prokaryotes. Yep, there we go. Those are all in prokaryotes. That's how prokaryotes increase genetic variation. Pause here to take notes. Woohoo! Favorite subject, biotech. I'm a biotech queen. I love biotech. Boston's the place to be in biotech. Let's talk about it. So, biotechnology is really just manipulating genes and being able to have them do different things. So, both DNA and RNA can be manipulated through genetic engineering. Fun fact, when I was in college, I really wanted to be a genetic engineer. I thought the idea of messing with genes to create something new was like really awesome. So gel electrophoresis, we're going to do this in the lab. It's a technique to separate DNA by fragment size. We load them into wells. So here you have what we call a gel. Up here you have the small wells. And an electrical current is applied. So the DNA moves from one side to the next, from negative to positive, because remember, DNA is negatively charged. And the thing that it's running through is kind of like a sponge almost, that larger DNA molecules get caught up at the beginning and then smaller get caught up at the end, so we can actually separate them by size. See, DNA fragments are negatively charged, so they move across towards the positive side. When I was in college, I accidentally put the positive where the negative went, and my DNA went that way. Gave me no results after hours and hours and hours of work. It was lovely. I'll remember it for the rest of my life. But we are going to do this technique in the lab. Pause here to take notes. All right, PCR. PCR is more commonly used in professional labs. It's called polymerase chain reaction. And it's to make several copies of a specific DNA sequence. This is how forensic DNA analysis happens. So you have an original piece of DNA over here that has to be replicated. And essentially the segments are amplified. So we have one segment here and we put it in with a bunch of nucleotides and a bunch of primers and like all the stuff we need to go through and create multiple copies of one piece of DNA. So this is how forensic investigators can take a small piece of DNA and just make more of it to be able to test it. Right. So once we see that, we can look at the results via electrophoresis, which separates out each of those lengths of DNA. We will not be doing this in the lab. We don't have the equipment to do this in the lab. But if you are working at Remix Therapeutics over the summer, you will be doing what's called qPCR, which is qualitative PCR. It's essentially measuring the amount from here to here. Good stuff. Pause here to take notes. Lastly, we have DNA sequencing. So DNA sequencing is the process of determining order of nucleotides in DNA. This is what the Human Genome Project did ad nauseum. So you can see what they do is they radio tag each individual one of the nucleotides, represented here with different colors, and they run it through what's called GCMS, gas spectrochromatography, GCMS, gas spectrometry mass something. It's all chemistry stuff. You can ask Mixon, she knows. And what we get is these peaks showing where each one of these base pairs is. So that's sort of how we sequence DNA. Pause here to take notes, and that's it. You finished Unit 6. Congratulations. I'll see you tomorrow.